Good morning, everybody. Or yes, good morning. I think it's morning in most places. Welcome to the webinar. We're going to give a minute here for people to trickle in, and then we'll go through our intro slides and start the speakers.
While we let people come in, I'll go ahead also and put up the links for the mailing list registration and speaker nomination forms if people would like to add anything. All right, so today we have two great speakers. Catherine Hoar will speak first, assistant professor at Boston College on fungal communities in wastewater. And Hannah Healy will speak second on bacterial contributions and biofilms in wastewater surveillance signal. I was a little late in putting together the slides, so we do have an excellent lineup of speakers for June, and keep an eye out for that announcement via email. But in the meantime, we'll go ahead and get started. And Catherine, if you'd like to go ahead and share your slides and get started when you're ready.
Okay. All right, can you see my slides okay?
Yeah, looks great.
Yes.
Okay, great. Well, thanks so much for the opportunity to share some of what we've been working on related to fungi in wastewater. So as Kyle mentioned, I'm an assistant professor at Boston College. I thought I'd just introduce myself a little bit. So I'm part of a new undergraduate engineering program at Boston College. It's a program in human-centered engineering. We're actually graduating our second class of seniors this month, which is exciting. So in this role, it's been a lot of fun to not only build a new lab, and I'll talk about some of the work that we've been working on, but also build a curriculum and a program where students are thinking from the beginning, from their first engineering course, how they can collaborate and think across disciplines to address some of our biggest challenges, which I think I just wanted to share that because I think it's really in the spirit of this RCN and our work generally in wastewater surveillance. So I also bring this up to highlight, as many of you I'm sure know, wastewater surveillance is such a great case study for the impact of engineering on public health and the importance of interdisciplinary considerations in engineering applications. And so centering our work and our engineering on ethics is something I think a lot about now. And in developing this program and in our coursework, we've attempted to think really intentionally, not only how can we engage undergraduate students meaningfully in research, but also challenge them to think about ethics and their role as engineers, and give them experience with interdisciplinary collaboration. So in particular, we've worked to foster sort of a more seamless connection between our engineering department and another new program here at BC, which is the Global Public Health for the Common Good program. And an example of some of this work is what I've done with Dr. Summer Hawkins, who's a professor in the public health program, actually the associate director, in her Contemporary Issues in Public Health course, where we've designed a unit and student dialogue around ethics of wastewater surveillance, drawing on a lot of the things that we've talked about through this webinar series and through other workshops and things and a lot of your work. So I just wanted to highlight some of the things that the undergraduate students have brought up when we do this. So they do some background reading. We talk about, some of the work that I've done. And I think as you can see, if you look at some of these quotes, these are just like general things that the students have brought up. They're thinking a lot about the things that we're thinking about in the field. So I just find this really encouraging. I like to share this because I think it gives me a lot of hope for the thoughtful engineers and public health professionals that are going to come up next. So if anyone wants to chat undergraduate engineering education and integration with public health, I'm here for it. But okay, sorry. Back to the topic for today. Why fungi? Why am I talking about fungal pathogens in wastewater? So pathogenic fungi infect billions of people every year, causing a range of diseases from minor skin afflictions to lethal brain infections. Estimates indicate that over 6.5 million people are affected each year by like an immediately life-threatening fungal disease, leading to over 3.75 million deaths. So I've got an infographic here from the CDC summarizing the burden of fungal diseases in the US. Despite their importance for human health, fungi remain chronically understudied, both as human pathogens and as resident members of wastewater microbial communities. In fact, if we look at studies that are looking to characterize the sewage microbiome, the focus is generally on bacteria, right? So when a paper says microbial communities, a lot of the time that's going to be focusing only on bacterial communities. Certainly we know more about viruses for sure, especially since the pandemic, but we have a much less of a better understanding of the spatial and temporal variations of non-bacterial communities in wastewater treatment and in wastewater treatment plants. So just sort of a rough estimate, thinking about what's out there right now, a little Web of Science topic search, looking for wastewater surveillance, wastewater-based epidemiology, and different types of microorganisms. The most is obviously on viruses, as you might expect, more coming up on bacteria, but fungi is really underrepresented. And so in the context of wastewater surveillance, even though fungi comprise a major portion of human pathogens, they've just received less attention. An exception to this, obviously, is C. auris, whatever you're calling it at the moment, which, as many of you know, is a highly pathogenic and resistant fungi. And so there has indeed been work successfully detecting C. auris in wastewater. However, as far as I know, no conclusive relationship has been established between levels of C. auris genetic material in wastewater and clinical cases. So this motivates one of our research directions, which is looking at how we can better characterize the wastewater microbiome. And in my talk, I'm going to highlight a lot of work from undergraduate students in my lab. My lab is made up of undergraduate students. And so Julia Littlefield is working on a literature review compiling studies that have looked at fungal communities in sewage, and also mining metagenomic data from studies, even if fungal communities weren't the focus of that study. So stay tuned for more on that. Of course, there are reasons why fungi are less studied in wastewater related to scope and interest, but also just generally fungi are understudied. And part of that is likely due to the difficulty in detecting especially their genetic material when applying methods that are typically applied and that have been optimized for bacteria and viruses. So fungi have this hardy, really dense cell wall. They're coated, and so they're harder to crack open. And they can also become dormant and do all these things that I'm not used to as someone who works with bacteria and viruses. So for this work, we've also teamed up with an expert in fungal biology, Dr. Zoe Hilbert, who's in the biology department here at Boston College. And so thinking about, hardy cell walls and having a hard time breaking fungal cells open, that's why lysis is so important, right? And this is probably nothing new to people who know fungi, but just a great little study to prove to ourselves. A couple undergrads in my lab looked at the impact of bead beating time on recovery of fungal DNA. So here they took influent wastewater, concentrated by centrifugation, and used a commercial kit with silica membrane column-based DNA extraction, and just tried with the lysis buffer, no vortexing, 10 minutes, 40 minutes, which is the recommended, and then 60 minutes. And then we quantified fungal DNA with qPCR targeting ITS. And so the importance of vortex time is pretty clear here, as we'd expect. More time definitely helps. I think one thing I'm not showing here, and we have to look into a bit more, is that this optimal time might actually be different for different fungal species. So we actually do see a little bit of a difference in this pattern, especially after that 40 minutes for different species. So bead beating time matters, lysis matters a lot. So to start thinking about how we will optimize DNA extraction methods, we've been starting with commercial nucleic acid extraction kits to see specifically how well they do with fungi. Some commercial kits do report recoveries of fungi or for certain fungal species, but many do not. So for these first experiments, we just spiked Saccharomyces cerevisiae in sterile water. And then Kat, an undergraduate student in my lab, and Leah, a research associate in the Hilbert lab, looked specifically at 4 different silica membrane spin column-based kits. And here we're looking at, these are just full DNA concentrations because we spiked in yeast here for each of the kits, A through D. And I think it was 6 replicates each here. Clearly we see 2 winners. We did double check that these kits C and D were getting some fungi out of there. So we did ITS targeted PCR, but just much lower performing kits. So not all kit is created equal. So then moving on with those winners, kits A and B on the left here, we tested the 2 best performing kits with wastewater from 3 different facilities in the greater Boston area. Again, 24-hour composite influence samples using centrifugation for concentration. So this is endogenous fungal DNA in wastewater samples, again, based on qPCR quantifying fungal DNA targeting the ITS region. So we don't see any clear trends between these 2 kits having higher yields over the other, but we are finding that across wastewater samples, DNA makes up anywhere between like 0.05 to 0.1% of the total extracted DNA. And so what we're trying to answer with these sort of methodical little studies is, is this representative of the fraction of fungal DNA in wastewater, or is it an artifact of extraction? So then we also wanted to see how extraction kits might impact the makeup of the communities that we are able to retrieve from these samples. So we did ITS sequencing, ITS2 sequencing on the same sample using DNA extracted from kits A and B, again, those 2 high-performing kits. And here I'm showing the fungal communities for that facility A, one of the facilities in the greater Boston area. And we are looking at relative frequency of the top 6 phyla and the top 6 genera, which happen to be in the same phylum here. And so we see also similar community profiles here, which is encouraging. We don't seem to get a bias from the extraction kit. And we also see similar alpha diversities, something we're going to assess as we look at each of the methods that we're working through. We've also started to look at fungal communities in different areas and over time. So again, this is another facility in the Boston area, just highlighting 2 community profiles from summer and winter samples. We have some minor differences over time, but fairly consistent dominant players at this facility. And the big standouts here, as expected, things like Candida and Saccharomyces. And so these are also genera that dominate in the hospital samples that we and our collaborators have been looking at. So for that work, we've been collaborating with researchers at the Children's Hospital of Philadelphia to look specifically at hospital wastewater, trying to compare the fungal communities and the pathogenic targets that we find in municipal water compared to hospital wastewater. And so their strategy, they're using different concentration here with a NovaPrep concentrating pipette, but also culturing with Chromagar, and then doing metagenomic sequencing along with qPCR. And so what we've found when we're comparing our municipal samples with the hospital wastewater samples is that in hospital samples, we're finding between 1% and 5% of total reads are fungal reads. So comparable, but maybe a bit more, at least based on fungal DNA mass, which is not maybe the best comparison, but definitely fungal DNA makes up a small portion of the total DNA. And then we also found that our diversity indices, alpha diversity in municipal wastewater is a bit higher than what we're finding in hospital wastewater, which is perhaps expected. So we're also interested in the fate of fungal targets in sewage and in sewage contaminated water. So thinking about decay and persistence of DNA signal across different fungi, something that we've looked at every time a new target comes up. And so here we're looking at concentrations based on genera-specific ITS qPCR for Candida species and Saccharomyces species in wastewater, so endogenous fungi, over time just left at 20 degrees C. And so it looks like, at least from this study, that decay of DNA signal varies for these different fungal targets, meaning that we can't just assume decay for one fungi is going to apply to others. So here are some results from a preliminary study we did looking at DNA signal from Candida species in wastewater and Saccharomyces species in wastewater after spiking with either Candida albicans or Saccharomyces cerevisiae. And so we did these tests at 4 degrees, 20 degrees, and 37 degrees, looking at decay over time. I think we're seeing mostly what we'd expect in terms of the influence of temperature. But when we compare what we're seeing for decay for each fungal species, it's not necessarily matching the decay of total fungi. So again, just being careful about how we think about decay of individual genera and species. I will say one caveat here is that we did obviously definitely had Candida albicans to start with. So we can't really get at DNA decay rates exactly from these kinds of experiments, but we're thinking about that. And our next step is also to think about viability. So we're working on this as well. Here we're just looking at total fungal DNA, again, from qPCR in wastewater that's just stored in the fridge. And so, taking a sample after about a month, we still see viable fungi present. Also, a whole lot of bacteria, which is another sort of troubleshooting thing that we've worked through. So you can see this plate is pretty crowded. So another thing that Leah has been working on in Dr. Hilbert's lab is optimizing our incubation temperatures for our plates to look at total fungal diversity in these wastewater samples, and also antibiotic concentrations. And so I think we've landed on 30 degrees C being probably the best temperature to still get fungal growth, with a combination of antibiotics to really make sure that we're getting rid of bacteria that's in there. And then another thing that Leah's been working on is thinking about looking at decay of viability of specific controls that we spike in. So we've been using some labeled Candida species that we're spiking in to try and track decay of viability over time. So with that, I'm reaching out now to see if anyone would like to work with us to help better characterize the wastewater microbiome. We're starting a database. If you'd like to contribute any sequencing data and metadata, definitely reach out. We're hoping that improving our detection methods will help us get a better sense of microbiomes of wastewater samples we get from across the country, and ultimately, along with the decay experiments, inform interpretation of surveillance data, and think about associated risk with not just sewage, but sewage polluted waters, CSOs, etc. And then I think I'm just about out of time, but one more thing I just wanted to highlight was, every time we think of a new varied target, I start to think, more methods. So I think, in addition to wanting to look for multiple targets and also make sure that we're having a normalization metric that is reasonable for the targets we're looking at, I think we need to keep thinking about multiplex detection. So I just want to, with that, highlight one collaboration with partners here at BC who've developed a device, an electric biosensor that might be one potential solution for getting at multiplex detection. And so this device, you can see it's real small up here. We've got an image. This is a graphene-based device, graphene-based biosensor that can detect multiple targets. Here they've created one that's able to look at RSV, flu, SARS-CoV-2, and caffeine. And the detection method is based on DNA aptamer probes with specific affinities for these targets. And when the target's attached to graphene, the graphene's quite sensitive to charge. And so measuring that change in charge and that shift in voltage as they apply. Oops, sorry, wrong slide. Yep. Shift in voltage as they apply. Sorry. Oh, okay, my animation's not working. But sorry, as the aptamer, as the target binds to the aptamer, you can detect that shift in resistance as the voltage is applied. Okay, cool. And what's cool about this is a potentially handheld device that could move us from, collecting samples on site, moving them to our lab and doing concentration and specific detection methods that are different for biological targets and chemical targets, and potentially doing this all in one handheld device. So potentially moving us towards point of need surveillance. And there is a new paper out where the team has been looking at how different strategies for measuring that shift in voltage could be used for lowering LOD or potentially just having better presence absence detection. Okay, sorry, I went a little bit over time, but I thought I wanted to thank collaborators both at BC and at CHOP, and I'm happy to take any questions.
Hi, thank you so much, Catherine, for that great presentation. Very clear and also perfect amount of time. We have a bunch of questions in the Q&A, and I'm gonna, since I'm moderating, I'll ask my question first, which is, how do you think the fungi get into the wastewater? Like, how are they shed? What do you think?
So things like Candida and Saccharomyces for sure are just like natural inhabitants of the gut and gastrointestinal. So that for sure. Things like Candida albicans, I don't know. I'd love to hear more from other people. Sorry, Candida auris, because that's not something that can proliferate in the gut, so far as I know.
Are there any studies of the concentration of those in like feces, for example?
Yes, I think for sure there are definitely some gut microbiome studies, but I don't about the pathogens like Candida auris, I don't think that we have in feces.
Let's see. There's a bunch of questions here in the chat. Some of them are really specific. So there's one question about your experiments. It says the last figure, but I'm not sure exactly what figure that was. The question is, was DNA spiked or was it an organism with target DNA? I think maybe it was, maybe not this one, but the one before these experiments, did you just spike nucleic acids or the organisms?
Whole cell, the organisms.
The whole cell. Okay. Let's see. Yeah, and then there's another question about the experiments is, did you obtain the fungal strains commercially available, or did you grow them up in-house to then spike them in, or both, I guess?
I mean, I guess they were grown in-house.
Okay.
Yeah.
And then there's a question of whether you plan to do any partitioning experiments between liquids and solids for the fungi.
Yeah, I mean, we're definitely going to look at settled solids versus influent. But lab-based sorption, I'm not sure. I think that would be tricky.
How big are they, the fungi?
That's a good question.
Are we gonna say fungi or fungi? Fungi.
Well, those is fungi, so maybe I should
fungi.
say
Okay. Yeah. So aren't they pretty big? Like, would they be
Yeah, I mean, they're
classified
gonna,
as a solid
yes,
anyway?
yes,
Yeah.
yeah.
Okay. Let's see. Uh, there's a question about like, you mentioned that different fungi could decay differently in wastewater, and I guess that's the nucleic acid target.
Mm.
Do you have any idea why? Like, what do you think? Have you thought about like reasons why they would be different? And do
Yeah,
you
I
want to
mean,
share with us?
sure, yeah. I mean, you can even see from some of the, I'm not prepared to say anything conclusive about this, but you can see some things kind of going up and down, at least with these preliminary experiments we've done with the graph that I'm on right now. I mean, fungi can do funky things like be dormant. I don't know if like hanging out in the wastewater for a little bit makes them more able to be detected or like
Mm,
it's easier to bust them open. Um, so yeah, I think there's a lot going on, and I think we'd need some very specific controlled experiments to think about that. I mean, I think the
mhm,
DNA itself, we haven't done like spiking in just raw DNA. I'm not sure if we'd see a huge difference if we spiked in extracted DNA, but I think it has more to do with what's going on with the fungi as they sit
mhm.
even in water, if it was
Yeah.
in water.
There's a question of whether the organisms you spiked in these experiments were, quote, dead or alive when you spike them in.
They were alive, yeah.
They were alive. Okay. Um, let's see. Um, I guess I was wondering, have you done any work to do assay design for the fungi? And if so, um, is it hard to design assays? Like, are their genomes like similar? Are they quite different? What are your thoughts on that?
Yeah, we've done a little bit of that. It is tricky. Like, for example, the assay we used here, it's not specifically for Canada albicans, and it's not for all Canada either. So I think that's something we definitely have to optimize a bit more. I think that's something,
Mhm.
um, yeah, still need to do work on that. But yeah, I think it's a little tricky.
Sorry, I'm like firing questions
No, it's
at
great.
you. I'm just sort of going off the questions from the crowd. So then someone, Kyle, actually, I can say who because he's right here. He says he's curious about the spore state, and
Yeah.
do you know how to make these fungi turn into spores, and have you spiked them as a spore versus not a spore, and could that affect things?
Yeah, that's a great question. I don't know. I have to talk to Zoe about that. I think sometimes it's just hard to know, especially if you're leaving them for a while.
Mhm.
But we haven't deliberately spiked in spore state, but it's a great question. I think we have to think about. Yeah.
I know, like, for Clostridium perfringens, right? Like
Mm.
they say if you pour boiling water on,
Mm.
like if you're doing membrane filtration, you pour boiling water, then only the spores would survive and grow. That's like in the standard method or
Yeah.
some method for Clostridium perfringens. Do you think there's a surrogate for fungi that people could use if they don't want to have to culture it? Like, is there a bacterial surrogate or no?
I don't know. I just think they behave so differently.
Hmm.
Yeah, I don't know. I mean, my hope is that I mean, there's definitely been control. I'm forgetting the specific species that others have used when looking at Canada auris in wastewater. So there definitely are like controls that are not pathogenic that people have used. Um, but I don't know if we know enough to say that they're good surrogates or
Hmm. Um, okay, well, we're mostly out of time, so I think we'll wrap it up, but I think I'm speaking on behalf of the entire community. Thank you for taking on this hard problem. And stick with it because it does sound very challenging, and that's probably why nobody has really done it. Because there's so many variables. So thank you for doing this, and thank you for your great presentation. And we will now pass it to Hannah, who will go next, and Kyle will moderate. Hannah. Thanks, Catherine.
Okay, can everybody see my screen okay?
Yeah.
Um, Great. Hi, everyone. Thank you so much for having me today and for attending this talk. I'm Hannah Greenwald Healy. I am an assistant professor of environmental health at Harvard Chan School of Public Health. I started my lab here just over a year ago, so I'm really excited to share what we've been working on. I'll also mention I'm just down the road from Catherine, so we have a very Boston day at the RCN today. And Catherine and I have been collaborating closely since I started, and I'll give a brief plug for some of what we've been working on together at the end of my presentation. But today I'm talking about biofilm contributions of bacterial pathogens and antimicrobial resistance genes. I'll talk about fungi a little bit to wastewater surveillance signal at the hospital scale. So although less common than viral targets, and I think Catherine did a great job showing just how much work has been done for viral targets in relation to bacterial and fungal targets, but wastewater surveillance has not been as commonly applied for bacterial and fungal targets, but is being increasingly utilized. So even prior to the pandemic, there were some examples of this for things like salmonella. And then since then, we've seen this surge of many other studies on this topic, where people have looked at antibiotic-resistant bacteria in wastewater, fungal targets like we heard about with C. auris, and then lots of studies of antibiotic resistance at the resistome level, and then also looking at more indicator specific ARGs. And these studies have been conducted at the international scale, sewershed scale, and then also specifically at hospital scale. And wastewater surveillance for these targets is especially impactful in hospitals. So healthcare-associated infections, or HAIs, account for an estimated 1.7 million infections and 99,000 associated deaths in the US, like just in the US each year. And infection prevention is even more essential as rising antimicrobial resistance, or AMR, makes these infections harder to treat. And so hospitals are very interested in approaches that could help them with this infection prevention, such as wastewater surveillance as an early warning. And so potential uses of wastewater surveillance at the hospital scale include that they could be sentinels of broader community health, or for HAIs, they can serve as early warning or confidence that outbreaks have been controlled and contained, and they could scale back some of their infection prevention measures. And then it could even help hospitals construct their antibiograms or monitor changing antibiotic resistance in the patient population. But notably for all of these applications, we want to understand what's coming from the patient population and not from environmental reservoirs. So there are some applications of wastewater surveillance where you might be interested in more of a One Health perspective, where you're interested in what's coming from humans, animals, and the environment. But specifically in this case, we really want to isolate what we're finding to what's coming from patients. And most of the causative agents of these HAIs are bacterial. So you can see here a distribution of the different species that are responsible, things like Klebsiella, Pseudomonas aeruginosa. You may have also heard of the escape pathogens, which are a group of highly virulent multi-drug resistant causes of HAIs. And notably, all of these pathogens shown on the slide have some level of biofilm formation potential. This is generally documented on tissues because they're causative agents of skin and soft tissue infections, or on medical devices, things like on catheters where they can cause UTIs, or ventilators where they can cause ventilator-associated pneumonias. But they have some level of biofilm formation potential. However, that their abundance in sewer biofilms specifically is not well characterized, but there could be potential for this environmental persistence and growth. And I'll note that some of these we have documented and others have documented to grow in sink drain biofilms for this study, and that was some of what prompted this work. And it matters if these are growing and persisting in our sewer biofilms because there tends to be a two-way exchange between microbes that grow in biofilms and their proximal bulk water. So this is something that is very well demonstrated in drinking water systems, and those are some of the diagrams I'm showing here, where organisms from the bulk water can seed biofilms where they grow and persist. And then they can go back into the bulk water through dispersal or sloughing mechanisms. And so if this were to happen in wastewater, we may see signal from sewer contributions and not just the patient population. If you want to note that our sewer pipes don't look like our drinking water pipes, they've just been less studied, so I don't have a good diagram on them. But in our sewer biofilms, or in our sewer pipes, we wouldn't have the pipe be fully submerged with liquid generally. So when I talk about these sewer biofilms, I'm talking about what this manuscript referred to as a slime layer. So the bottom part of the pipe that would be in contact with the sewage. There's also settled solids in our sewer pipes, more so than in drinking water systems. And then if you do look up any literature on biofilms and sewer pipes, what you'd probably find mostly is description of what, again, this publication called the moisture film, which is a biofilm that can form on the top part of the pipes. And we're generally concerned with those because sulfur oxidizing bacteria can cause corrosion of pipe materials. But again, today I'm focusing on biofilms that have the potential to exchange with the sewage. And so we had some research objectives for the study. We wanted to characterize the extent to which these sewer biofilms could reflect and reshape wastewater pathogen and AMR signals. We first wanted to characterize sewer biofilm microbial communities, because there really haven't been prior in situ studies of native pathogenic taxa in sewer biofilms beyond sink drains. We sought to estimate bacterial community contributions from biofilms to wastewater, and then identify wastewater pathogens and ARGs that were likely derived from plumbing biofilms. And so we conducted a 4-month study at Yale New Haven Hospital in collaboration with the Peccia Lab and the Turner Lab at Yale, and then also Perimeter, previously known as Ginkgo Biosecurity. The study is being led by my postdoc, Amanda Darling, and I'll note that sampling was conducted across 5 wards in the hospital, so technically this is a ward scale surveillance study. We collected various types of samples. So of course, wastewater, we collected this with composite autosamplers at 45 different time points over the study at these 5 locations. We connected the autosamplers directly to branch drains, so these horizontal pipes that were points of convergence for all of the rooms on the ward, sometimes spanning multiple floors. And then we went into those branch drains to collect biofilms from the same location where the wastewater sample was being collected. And you can see here the end cap gets pretty full of sediment, so we would open that up and kind of clear that out, and then stick a long dowel into the pipe so that we were actually hitting the point where the wastewater was flowing. We were able to collect these at 3 time points over the course of the study at each of our locations. And then we also collected tap water and sink drain pipe biofilms from many fixtures upstream of our collection points. And we used 2 types of sequencing for this study. Many of you will probably be familiar with 16S RNA gene amplicon sequencing. We sequenced the V4 hypervariable region. This enables broad characterization of bacterial communities where we're sequencing these specific amplicons. A little bit different and maybe a little bit newer technology. We used hybrid capture-based enrichment or probe capture-based enrichment. Many of you may be familiar with the viral surveillance panel from Illumina. In this study, we utilized the RPIT panel from Illumina, which contains probes for 180 bacterial pathogens, 50 fungal pathogens, 40 viral pathogens, and 1,200 ARGs. And this works where you can have thousands of these biotin labeled probes that would attach semi-selectively to your targets of interest. You can then capture those fragments using streptavidin magnetic beads and wash away some of the background. So it gives you some enrichment over typical shotgun metagenomics, but it enables broader capture than an amplicon sequencing approach might, and it's a little less selective. And then also, if your probes are designed correctly, you can get species level identification. And for our bioinformatic pipelines, for 16S, we use the Data2 pipeline. For probe capture metagenomics, we had a read-based analysis pipeline where after QC, we used Kraken 2 with our chosen quality control thresholds. And then for ARGs, we used Diamond to align to the card database. And although this is a relatively simple pipeline, this was actually quite an iterative process where we had to think through which profilers or classifiers would be able to work with this type of probe capture enrichment sequencing where we aren't getting full genome coverage. And so certain marker-based profilers are incompatible with the probes on the panel. We were also able to use some context of other studies that we had done using these sample types where we had culture context to see things that were maybe too conservative. And then we had clinical context where we knew that we should not be seeing certain rare pathogens that were not detected during this time period by the hospital, but would have been in retrospect, to look at some classifiers that were too permissive. And then we used BLAST to kind of confirm that our approach was working. And so looking at these sewer biofilms, they were much more diverse than tap water and then our sink drain biofilms. They had a comparable diversity to our wastewater samples and had significant overlap with our wastewater samples. So looking at the NMDS, we have our tap water and then our sink drain biofilms, and then our sewer biofilms and wastewater are over here. They were statistically distinct communities. There is a 50% overlap in taxa at the genus level. And so as we start to estimate the biofilm contributions to the wastewater microbiomes, we used feast source tracking to estimate the percent contribution of each of our upstream sources to wastewater microbial communities as a whole. And so we found that tap water and sink biofilms had negligible contributions. The majority of the contributions were from an unknown source, which we think would be the human population. And then, but we did see some contributions from these sewer biofilms. So this is at the ward scale, but averaged across the wards, we estimate 9% of wastewater bacteria were derived from sewer biofilms. We can also look at specific pathogens that were detected across these environments. So here I'm just showing pathogens that were detected in at least 3 sewer biofilm samples, because of course there could be two-way exchange. And we think that if something was just detected once in a sewer biofilm, it could have been just transient deposition from wastewater to the outer surface of the film that we pick up with sampling. So we're limiting it to just 3 or more detections. But we still see many clinically relevant pathogens showing up in our sewer biofilms, shown in green, and then even in some sink biofilms, shown in this reddish color. Things like Pseudomonas aeruginosa, Klebsiella pneumoniae, Stenotrophomonas multifida point out again that all of these or most of these have some prior documented evidence of biofilm formation or even have direct evidence of persistence in plumbing biofilms. And so none of this is too surprising. But it does support that many clinically relevant pathogens that would be wastewater surveillance candidates and that the hospital was really interested in monitoring were consistently detected in sewer biofilm, supporting there could be potential environmental contributions obscuring caseloads. And this is not a new concept. I mean, it doesn't get mentioned, I think, as much as it should in a lot of papers on these types of targets. But notably, there's this paper that I discovered somewhat recently, but was published in 2016, where they conducted wastewater surveillance at the hospital scale of carbapenemase producing Enterobacter ACA. And they concluded that the wastewater signal was totally unrelated to the clinical isolates, and they proposed that this was likely coming from growth in sewer and in sewer biofilms and sediments. And so they weren't able to directly study the biofilms and sediments in their study, but we think our work here helps support this framework. We can also look at the relative abundance and kind of temporal and geospatial stability of these pathogens that we were observing. So Pseudomonas aeruginosa was the most abundant pathogen, both in wastewater and in sewer biofilms across all of our wards. It had fairly stable concentrations over the different time points of the study. And then you can see distributions of all of these pathogens over here, where again, the distributions look fairly consistent across the wards and between wastewater and sewer biofilms. I do want to note that some pathogens were detected in wastewater, but not in biofilms. So some of these included Bordetella species like Bordetella pertussis, different species of strep, Fusarium, which is fungal. And then, of course, there were also many pathogens that were not detected in our study in any matrix. So things like C. auris. And then in addition to pathogens, we saw substantial overlap in antibiotic resistance genes between sewer biofilms and wastewater. So this is an upset plot where we're showing the detection across multiple environments of specific genes, and then they're colored at the drug class level. So in this first column, we're showing all the genes that were observed both in wastewater and in sewer biofilm samples. The third column shows ones that were detected in all three environments. So 66% or over 1,500 unique ARGs were detected in both sewer biofilms and wastewater, highlighting the potential for environmental contributions. And then if we zoom in on specific genes that are commonly used as indicators or commonly observed in some of these prior wastewater environmental studies, we see that there's a lot of overlap in these as well, and just very consistent detection in sewer biofilms and then even in the drain biofilms that are upstream. And so while this is still very important, again, from a One Health perspective, that we're propagating all of this antimicrobial resistance in the healthcare setting, it again highlights that we might not be able to say that this is really coming from the patient population as we conduct these studies. We conducted one more analysis, kind of looking at preferential enrichment of these genes across the different environments, so between biofilms and wastewater. And we found many genes that were enriched on a relative abundance basis in biofilms. These included many resistance genes that are associated with environmental opportunistic pathogens. And it also included some resistance genes like CAC-L, which confers resistance to quaternary ammonium compounds, which would be a surface disinfectant. And so this would have environmental origin because it's not something that it's not an antibiotic that people take. And so this is further demonstrating that biofilms are hotspots for resistance genes that are of environmental origin and not necessarily of human origin. And so takeaways, sewer plumbing biofilms represent a persistent and diverse environment that harbors clinically relevant pathogens and AMR genes. We further support here that wastewater likely reflects both human and environmental inputs. And we hope wastewater surveillance studies will acknowledge background levels and non-human signal. There are a few ways we might be able to do this, maybe just starting by avoiding poorly suited targets, or hopefully adjusting for baseline levels of these organisms, but we need more work in that area. And the next step for our team is we're hopefully very close to getting the paired clinical microbiology data from this time period so that we can do more work relating the temporal and spatial trends in the wastewater to the clinical data. We think some future directions for this work could include bench scale experiments to better estimate exchange rates and directionality. More sewer biofilm studies, including of mains, so like in the actual sewer system, because this was still building scale. And then potential for these thresholding approaches. And then before I end, I want to plug an upcoming publication. So this is a study that Catherine and I co-led with contributions from a fantastic team of researchers working in this related area. But we have a perspective piece on how we think wastewater surveillance can inform cancer epidemiology and evidence-based interventions. And so we hope that you'll check that out when that's officially online. With that, thank you so much for attending. Thank you to all of my amazing co-authors, Amanda Darling for leading this work, and all of our collaborators in the H2O lab. Happy to take any questions, and here's my email for any follow-ups.
Great. Thank you, Hannah. Very, very nice presentation. Reminder for folks, I'm seeing some questions come in, but if you do have questions, go ahead and put it in the Q&A box and you can upvote if you have a specific question that you want answered. Selfishly, because I have the mic, I'm going to ask my own question first. In this study, you were looking at sort of building ward level biofilms. Do you think the story would change if you were able to sample several miles of a city sewer or something upstream of a wastewater treatment plant? Or do you think that it would be a pretty similar sort of conclusion of about 10% biofilm derived? Just your thoughts on that.
I think that's a great question. I think it's likely to change, but I don't know in which direction because, I mean, as you get deeper into a sewer system and the wastewater has traveled over more pipe surface area, you could have more contributions just because of the more opportunities for exchange with the biofilm. And so in that case, I would say maybe biofilm contributions could go up. But there's also a chance that the relative proportion might go down if we were seeing more consistent wastewater flows from the human population. So I'll note that because this was such a small population size and such a small area, I mean, this could be some of these wards, it's maybe like 50 people contributing. I think there's maybe a little bit lower biomass in general than in municipal wastewater, and therefore the fraction of contributions from the environment could go up.
Yeah, it's very interesting. Really, really great start. Allie was asking, how did you sample the sewer biofilm? So did you use a specific tool? Where in the pipe were you scraping the biofilm? Some more details on that.
So we used a swab, so just like a flocked cotton swab, but we attached it to a long wooden dowel, so that we could actually insert it into the pipe at the point where wastewater was flowing, rather than like getting closer to the end cap, where you would expect some sediment buildup, but not actual exchange with the wastewater. And we only swabbed the bottom half of the circumference of the pipe, where we would expect there again to be like some flow over that surface area.
Great, thank you. Can you comment on the challenges or lessons learned when using the Illumina RPIP panel?
Yeah, I mean it's definitely different than shotgun. I think we realized compared with some of these other probe capture enrichment panels like BSP that gives you probes covering the entire genomes of the target you're looking for. I think one of the challenges here is that this panel includes targets for a very small fraction of the genomes. And so you can't reconstruct mags of these organisms. It kind of restricts you to read-based analysis and doesn't really enable things like strain-resolved sequencing. And so that was a challenge, realizing that like marker gene-based methods won't necessarily align with the regions that you've enriched for was a challenge. So maybe there could be some work, like if there were more custom panels being designed to align with marker-based methods. Um, and Oh, and then also, so this panel, I think it's very appealing because it has so many targets on it. It seems like, wow, it's really a one-stop shop to be able to detect all of these various things. But the more breadth that you have in your enrichment, the less depth that you're going to get. And so we weren't able to see still some of the more rare targets, notably viral targets that are included in the panel. And so if you want to do viral sequencing, you might still want to work with a panel that's more specifically looking for those targets rather than the breadth of this one.
Yeah, that makes sense. And I guess there's a follow-up question sort of related. Can you elaborate on Metaflan not being compatible with RPIP?
Yeah, so Metaflan is based on marker genes, and so they're looking at specific parts of bacterial genomes that could help with the identification. Um, the RPIP panel has probes designed for also specific parts of these organisms, but it's proprietary. You don't actually know exactly which parts you're enriching for, but it's not consistently the same marker genes that are included in Metaflan. And so we actually did try it just to see what our capture would be like. And we were still able to get taxonomic identification with Metaflan of some of the bacterial taxa that we expected to see, but it was very conservative. We had a very, very short list of things that it was able to pull out, like maybe less than 10 taxa. And we realized that we were probably missing a lot just because of a misalignment of the marker genes.
All right. In applied surveillance settings, do you see 16S RNA sequencing as sufficient for biofilm organism detection? Or would you need metagenomics or some other more detailed approach?
I think if you're thinking about like pathogen surveillance and health-based surveillance, you would want a more detailed approach. We only used 16S here because there had been so little work characterizing the broader community in these sewer biofilms, and because we were interested in more source tracking approaches of the whole community to try to characterize that exchange and that transfer of all organisms. But I think in any ongoing like health relevant surveillance, 16S would probably not be a good tool. It often can't, especially with just sequencing hypervariable regions, you often can't get below the genus level with taxonomy. Full length 16S could help.
Yeah, that makes sense. Um, Cara Nelson asks, if a hospital was interested in, adopting wastewater surveillance, do you think there could be some approach of scouring out the biofilm from sewers or flushing it out so that what you're getting is actually patient signal?
Interesting. I don't think that we'll be able to scour out the biofilm from all of these pipes. Um, we so uh, right before, like leading into this study, we had been doing a lot of work on sink drain biofilms at the hospital, where and trying to just remove those, which are a lot easier to access than pipes deeper into the building. And even with those, I mean, biofilms regrow on the order of days, so you'd have to be scouring your entire system and trying to remove biofilms like every few days for that to be feasible. But I do think that because of the stability that we saw in the signal across locations and over time, I think that maybe more like quantitative thresholding might be easier than like physical removal of the biofilm. You'd have to note that like if you saw any big increase in your signal above a threshold, it still could be due to environmental interactions like temperature changes or changes in infection prevention measures where different disinfectants are getting into your system. But I think that's what I think is the most promising approach for still being able to apply this.
Great. Thank you so much, Hannah. I wasn't able to get to quite all the questions. A bunch came in towards the end, but I'm sure that Hannah would be glad to follow up over email if people had specific questions as well.
Yeah,
So thank you
absolutely.
again to Catherine and Hannah for some great presentations, and keep an eye out for the announcement of our June webinar, which will be the first Friday of June. We'll see everybody in a month.